Hello there, and welcome to Nighttime Science, where the mysteries of our world unfold at the pace of falling snow, where knowledge drifts gently through the darkness like bioluminescence in deep water. Tonight we are descending into a realm that challenges everything we thought we knew about the boundaries of life itself. We are traveling to the ocean's deepest reaches, to pressures that could crush steel, to temperatures that hover just above freezing, to zones where oxygen barely exists and sunlight has never penetrated. In these impossible places, life not only survives but thrives in ways that seem to defy the very laws of biology. There are creatures down there that can reverse the aging process and become young again. There are microscopic animals that can survive the vacuum of space, temperatures near absolute zero, and radiation levels that would be instantly lethal to almost any other living thing. There are worms that have no mouths, no digestive systems, yet grow to lengths of 3 meters in complete darkness. There are cephalopods that live where there is almost no oxygen, feeding on the debris that falls like snow from the sunlit world above. These are not myths or legends. These are real organisms discovered by scientists who ventured into the last truly unexplored frontier on our planet. And their existence forces us to reconsider what we mean when we say something is impossible. Because if life can flourish in these extreme environments, then perhaps the universe holds far more possibilities than we ever imagined. So settle in, let your breath slow to the rhythm of deep ocean currents, and prepare to explore the organisms that shouldn't exist, but do. Before we begin our descent, if you find comfort in these gentle explorations of science, perhaps consider liking this video or subscribing to the channel. It helps this quiet corner of knowledge reach others who seek calm understanding in the darkness. Now let us begin our journey into the impossible. The jellyfish that reversed death. There is a creature swimming in the Mediterranean Sea and in waters around the world that has discovered something extraordinary. It has found a way to cheat death itself, to turn back the clock of its own existence and become young again. This is not science fiction. This is Turritopsis dohrnii, the immortal jellyfish, and its story begins with a discovery made quite by accident. In the late 1980s, two scientists named Giorgio Bavistrello and Christian Sommer were studying jellyfish in laboratory tanks. They were keeping specimens of what they thought was an ordinary species, observing their life cycles, taking notes on their development. But then they noticed something that shouldn't have been possible. Instead of dying after reproduction as jellyfish normally do, some of their specimens were doing something else entirely. They were shrinking. They were losing their bell shape, their tentacles retracting, their bodies collapsing inward into small blob-like cysts. At first, the researchers thought they were simply watching their specimens die, but they continued to observe. And over the next day or two, those blob-like cysts began to change again. They began to develop stalks. They began to form the characteristic shape of a polyp, the juvenile stage of the jellyfish life cycle. The jellyfish weren't dying. They were aging in reverse. This discovery, presented at a scientific workshop in 1991, captured the attention of marine biologists around the world. Because what Bavistrello and Sommer had witnessed was something called ontogeny reversal, the ability of a mature organism to revert to an earlier stage of its life cycle. And Turritopsis dohrnii wasn't just doing this before reaching sexual maturity, as a few other jellyfish species had been observed to do. It was doing it after reproduction, after reaching full adulthood. It was, in effect, growing young again. To understand how remarkable this is, we need to understand the normal jellyfish life cycle. It begins with a fertilized egg that develops into a free-swimming larva called a planula. This tiny creature drifts through the water until it finds a suitable surface to settle on, usually the seafloor or the hull of a ship. Once settled, it transforms into a polyp, a stationary form that looks somewhat like a small sea anemone. This polyp can reproduce asexually, budding off more polyps to form a colony. Eventually, these polyps release free-swimming medusae, the bell-shaped form we recognize as a jellyfish. The medusa grows, matures, reproduces sexually by releasing sperm and eggs into the water, and then normally dies. But Turritopsis dohrnii has discovered a loophole in this one-way progression. When faced with stress, whether from physical damage, starvation, changes in water temperature, or simply old age, the adult medusa can reverse course. It sinks to the seafloor, its bell collapses, and it enters a transformation process that science calls transdifferentiation. Transdifferentiation is one of the most fascinating processes in biology. It is the direct conversion of one type of differentiated cell into another, completely different type of cell. Think of it this way. In a normal animal, once a cell has become specialized, once it has committed to being a muscle cell or a nerve cell or a skin cell, that commitment is permanent. The cell has turned on certain genes and turned off others, locking itself into a specific identity and function. But in Turritopsis dohrnii, when the medusa undergoes this reverse transformation, its specialized cells abandon their identities. Muscle cells become something else. Nerve cells transform. The cells of the bell and tentacles reorganize themselves entirely. The process is remarkably rapid. Within 24 to 36 hours, the collapsed medusa has become a polyp, effectively jumping backwards over several stages of its life cycle. And once in this polyp form, it can begin the cycle anew, eventually budding off new medusae that will grow to adulthood once again. This means that, theoretically, an individual Turritopsis dohrnii could cycle through this process indefinitely. It could live forever, never dying of old age, constantly renewing itself. This is why it earned its common name, the immortal jellyfish. But before we imagine oceans teeming with ancient, undying jellyfish, we need to understand the reality of this immortality. The key word is theoretically, because while the jellyfish can reverse its aging process, it cannot escape the other dangers of ocean life. It can still be eaten by predators. It can still succumb to disease. It can still be injured in ways that prevent transformation. In the wild, most Turritopsis dohrnii likely never get the chance to undergo their remarkable rejuvenation. They are consumed by fish or turtles or other predators while still in their medusa stage. But the fact that they have this ability at all has profound implications for science. Researchers studying these jellyfish have identified specific genes and molecular pathways that are activated during the transformation. They have found that genes involved in DNA replication, repair, and telomere maintenance are highly expressed. Telomeres are the protective caps on the ends of chromosomes that normally shorten as we age. Turritopsis dohrnii appears to have mechanisms for maintaining or even lengthening these telomeres during its reverse development. There are also genes involved in stem cell pluripotency that become active. Pluripotency is the ability of a cell to develop into any other type of cell, a property normally found only in embryonic stem cells. The immortal jellyfish seems to be able to reactivate this embryonic state in its adult cells. Scientists are now working to understand these mechanisms in detail because they hold potential clues for human medicine. Could we learn to regenerate damaged tissues by inducing transdifferentiation in our own cells? Could we find ways to reset cellular aging, to treat age-related diseases, perhaps even to extend human lifespan? These are questions that seemed like pure fantasy until we discovered that a tiny jellyfish, no larger than a fingernail, was already doing something similar. The immortal jellyfish reminds us that nature has been conducting experiments in biology for billions of years, far longer than human science has existed. And sometimes in the most unexpected organisms, we find solutions to problems we thought were unsolvable. We find evidence that death itself, at least cellular death from aging, is not necessarily inevitable. We find that the boundaries we perceive are sometimes just limitations of our imagination. Turritopsis dohrnii drifts through the ocean, small and translucent, easily overlooked. But within its tiny bell-shaped body, it carries one of nature's most profound secrets: the knowledge that time, at least biological time, can sometimes flow backward. That renewal is possible even after the journey seems complete. That endings in nature are not always final. The creatures that survive the void of space. If you were to venture into your garden and examine a handful of moss, looking at it under a microscope with enough magnification, you would likely find yourself face to face with one of the most indestructible creatures on Earth. It looks like a microscopic bear, plump and eight-legged, moving with a slow, almost clumsy gait. It is called a tardigrade, though it is more commonly known as a water bear, and it possesses abilities that seem impossible even by the standards of science fiction. Tardigrades can survive being frozen to temperatures approaching absolute zero, down to -272 degrees Celsius. They can endure heat above the boiling point of water, up to 149 degrees Celsius, at least for short periods. They can withstand pressures 6 times greater than those found in the deepest ocean trenches. They can survive radiation doses hundreds of times higher than what would kill a human being. And most remarkably, they can survive in the vacuum of space, exposed to cosmic radiation and the complete absence of air, for extended periods of time. These are not theoretical limits discovered in laboratory experiments designed to push boundaries. These are conditions that tardigrades have actually survived. In 2007, the European Space Agency sent tardigrades to space aboard the Photon M3 mission. The tiny creatures were placed outside the spacecraft, fully exposed to the vacuum of space and solar radiation for 10 days. When they were returned to Earth and given water, many of them reanimated and resumed their normal activities. Some even successfully reproduced afterward. This extraordinary resilience has made tardigrades subjects of intense scientific study. Because the question that naturally arises is how. How can an organism survive conditions that destroy almost everything else? The answer lies in a remarkable survival strategy called cryptobiosis, specifically a form known as anhydrobiosis, which means life without water. When tardigrades are faced with environmental conditions that threaten their survival, particularly the loss of water from their habitat, they undergo a dramatic transformation. They retract their 8 legs and their head into their body. They expel almost all the water from their cells, losing up to 97% of their body moisture. And they curl up into a desiccated form called a ton. In the ton state, a tardigrade's metabolism slows to an infinitesimal fraction of its normal rate, perhaps to as little as 0.01%. For all practical purposes, the creature is not alive in any conventional sense. It is not breathing, not eating, not growing, not reproducing, not repairing damage to its cells. All the processes we associate with life have stopped. The tardigrade has entered a state of suspended animation that can last for years, even decades. But here is what makes this truly remarkable. When water becomes available again, the tardigrade can reverse the process. It absorbs moisture, its cells rehydrate, its metabolism restarts, and within hours it is moving and feeding again as if nothing had happened. It has essentially come back from the dead, though technically it was never fully dead, merely in a state so close to death that the distinction becomes philosophical. What allows tardigrades to do this? The answer involves several sophisticated molecular mechanisms. One of the most important is a protein called DZSUP, short for damage suppressor. This protein was discovered in one of the most resilient tardigrade species, Rhamazottius variornatus. DSUP binds to the tardigrade's DNA and physically shields it from damage. When the tardigrade is exposed to radiation, which normally shatters DNA strands, the DSUP protein acts like a protective shield, preventing or reducing the damage. Even more remarkably, when scientists introduced this protein into human cells in the laboratory, those cells showed increased resistance to radiation damage. Another crucial molecule is trehalose, a sugar that accumulates in tardigrade cells as they enter cryptobiosis. Trehalose has unusual properties. It can replace water molecules in the cell, stabilizing proteins and membranes that would otherwise collapse when dehydrated. It essentially acts as a preservative, keeping cellular structures intact even in the complete absence of water. This is similar to how some seeds can remain viable for years in a dried state, though tardigrades have refined the process to an extraordinary degree. There are also heat shock proteins, molecules that help other proteins maintain their proper shape under stressful conditions. And there are highly efficient DNA repair systems that can fix damage when the tardigrade reanimates. All of these mechanisms work together to allow the tardigrade to survive conditions that would be instantly lethal to most organisms. But perhaps the most important thing to understand about tardigrades is that they are not extremophiles. This is a crucial distinction. Extremophiles are organisms that thrive in extreme environments. They have adapted to live in hot springs, in acidic pools, and highly saline lakes. And they actually require those conditions to survive. Tardigrades are different. They do not thrive in extreme conditions, they endure them. Their natural habitat is the thin film of water that clings to moss and lichens. They feed on plant cells and bacteria. They live in relatively benign environments. But those environments are temporary. Moss dries out. Lichens are exposed to harsh sunlight and cold. And so tardigrades evolved the ability to shut down, to wait out the bad times, to survive until conditions improve. They are not adapted to live in space or in boiling water or frozen in ice. They are adapted to not die when exposed to those conditions, which is a fundamentally different survival strategy. This distinction has important implications. It means that tardigrades die if exposed to extreme conditions for too long. Their chances of mortality increase the longer they remain in an extreme state. They are incredibly tough but not invulnerable. It also means that the mechanisms they use to survive are general-purpose protection systems rather than specialized adaptations to specific extreme environments. There are more than 1,000 known species of tardigrades found in nearly every habitat on Earth. They live on mountaintops and in deep ocean sediments. They inhabit tropical rainforests and Antarctic ice. They are in soil and sand, in fresh water and in the sea. Their ubiquity is a testament to their resilience. Yet for all their indestructibility, tardigrades are fragile in one specific way. They can be easily killed by physical trauma. If you crush a tardigrade, it dies. Its remarkable molecular defenses do not protect it from mechanical damage. This is perhaps fitting. It suggests that evolution does not create perfect organisms, only organisms that are well adapted to the specific challenges they face. For tardigrades, the challenge has been dealing with the periodic loss of their aquatic microhabitat. The solution was not armor or speed or intelligence, but the ability to simply stop living until circumstances improved. In recent years, scientists have begun to explore potential applications of tardigrade biology. Could we use DSUP proteins to protect against radiation damage in humans? Perhaps for cancer treatment or for protecting astronauts on long space missions? Could we learn to preserve cells or tissues using tardigrade dehydration techniques, improving organ transplantation or blood storage? Could we develop crops that can survive drought by borrowing genetic mechanisms from tardigrades? These are not idle questions. They are active areas of research. Because tardigrades, these microscopic creatures that look like chubby bears, have solved problems that human technology has not. They have found ways to protect and preserve life itself under conditions that we would consider utterly impossible. And in doing so, they remind us that nature is often the best engineer, that billions of years of evolution can produce solutions more elegant and effective than anything we might design from scratch. As you drift towards sleep tonight, you might consider that there are tardigrades in the moss outside your window, going about their slow, patient lives, carrying within their microscopic bodies the secrets to surviving the impossible. They are proof that life, when given enough time and enough challenges, can find ways to persist that exceed our wildest imaginings. Life without light in the crushing deep. In 1977, a team of scientists aboard the research submersible Alvin were exploring the Galapagos Rift, a region of the Pacific Ocean floor where tectonic plates pull apart and volcanic activity shapes the seafloor. They were nearly 2,500 meters below the surface, in a realm of complete darkness, crushing pressure, and near-freezing temperatures. According to everything science understood at the time, this should have been a barren wasteland, a lifeless desert of rock and sediment. But when Alvin's light swept across the seafloor, the scientists saw something that left them momentarily speechless. There were animals everywhere. Not just a few scattered creatures eking out a marginal existence, but dense, thriving communities. There were clusters of white clams the size of dinner plates. There were crabs scuttling across the rocks. And towering above it all, swaying gently in the deep-sea currents, were masses of giant tube worms, their brilliant red plumes extending from long white tubes anchored to the volcanic rock. The worms were enormous, some reaching lengths of nearly 3 meters. And they were growing in such dense colonies that they looked almost like underwater forests, their tubes packed tightly together, their red gills waving in the water like crimson flowers. The scientists had discovered hydrothermal vents, and more importantly, they had discovered that life could thrive in an environment completely independent of sunlight, powered by an energy source that had nothing to do with photosynthesis. The worms they found would later be named Riftia pachyptila, and they would revolutionize our understanding of where and how life can exist. Because these worms are strange in ways that go far beyond their size. Adult Riftia pachyptila have no mouth. They have no stomach, they have no digestive system of any kind, they cannot eat in any conventional sense. And yet they grow at astonishing rates, some adding 85 centimeters of length in a single year, making them among the fastest growing invertebrates known to science. So how do they survive? How does an animal with no way to eat obtain the energy it needs to grow? The answer lies in a remarkable symbiotic relationship. Inside each tube worm, in a specialized organ called the trophosome that fills much of its body cavity, there live billions of bacteria. These bacteria are not parasites, they are partners, and they provide the worm with all the nutrition it needs. But they do it in a way that was almost unknown to science before the discovery of hydrothermal vents. They do it through a process called chemosynthesis. Photosynthesis, which powers almost all life on Earth's surface, uses energy from sunlight to convert carbon dioxide and water into sugars. It is the foundation of nearly every food chain, the process that plants and algae use to create organic matter from inorganic compounds. But in the deep ocean, where sunlight never penetrates, photosynthesis is impossible. Chemosynthesis is different. It uses energy not from light, but from chemical reactions. Specifically, the bacteria inside Riftia pachyptila oxidize hydrogen sulfide, a toxic gas that spews from hydrothermal vents. In the process, they release energy which they use to convert carbon dioxide into organic compounds, primarily sugars and amino acids. These compounds then serve as food for the worm. This process is elegant and efficient, but it requires the worm to solve a significant logistical problem. It needs to provide its symbiotic bacteria with both hydrogen sulfide and oxygen, two chemicals that do not normally exist together because they react with each other. Hydrogen sulfide emerges from the vents in superheated oxygen-free water. Oxygen is present in the cold seawater surrounding the vents but is absent in the vent fluid itself. So the worm must simultaneously gather both substances from different sources and keep them separated until they reach the bacteria in the trophosome. Riftia pachyptila solves this problem with remarkable sophistication. At the top of its white tube, it extends a brilliant red plume of gill-like structures. This plume is red because it is filled with a specialized hemoglobin, a molecule related to the hemoglobin in our own blood but with some crucial differences. The worm's hemoglobin can bind oxygen from the seawater while simultaneously binding hydrogen sulfide, keeping the two separated in different binding sites on the molecule. It then transports both chemicals through the worm's circulatory system to the trophosome, where the bacteria can use them for chemosynthesis. This is a delicate balance. Hydrogen sulfide is toxic to most animals, including the worm itself in high concentrations. But the specialized hemoglobin and other proteins in Riftia pachyptila can handle it safely, delivering it to the bacteria without harming the worm's own tissues. The tube that protects each worm is equally remarkable. It is made of chitin, the same material that forms insect exoskeletons, but the worm's tube is specially formulated to be resistant to degradation. In the harsh, chemical-rich environment of a hydrothermal vent where the water can be acidic and filled with dissolved minerals, the tubes remain intact for years. The worms continuously add to their tubes at both the top and bottom, allowing them to grow and adjust their position as conditions change. Tube worms begin their lives as free-swimming larvae, drifting in the deep ocean currents. These larvae have no bacteria yet. They feed on yolk reserves and they have a rudimentary digestive system. But when they settle on a hydrothermal vent site and begin to transform into their adult form, something remarkable happens. bacteria from the surrounding water enter the larva through specialized tissues. Once inside, they migrate to the developing trophosome, where they take up permanent residence. As the transformation completes, the worm's digestive system degenerates and disappears, and the bacteria become the worm's sole source of nutrition. This dependence on bacteria is absolute. A tube worm without its symbionts would starve, but the relationship is mutually beneficial. The bacteria gain a stable, protected environment with a constant supply of the chemicals they need. The worm gains a reliable food source in an environment where conventional food is scarce. Hydrothermal vents are ephemeral habitats. They can be active for years or even decades, but eventually the volcanic and tectonic activity that powers them shifts elsewhere and the vents shut down. When this happens, the chemosynthetic communities die. The tube worms, deprived of their chemical fuel, cannot survive. But before the vents close, the worms reproduce, releasing sperm and eggs into the water. The fertilized eggs develop into larvae that drift in the currents, potentially for months or even years, until they find a new active vent where they can settle and the cycle can begin again. Scientists have since discovered hydrothermal vents in oceans around the world, and each location hosts its own distinct community of organisms. There are vents where clams dominate instead of tube worms. There are vents with specialized shrimp that harbor chemosynthetic bacteria on their bodies. There are vents colonized by unusual worms, crabs, and fish found nowhere else on Earth. More recently, researchers have found chemosynthetic communities in even more extreme environments. In the Hadal zone, the deepest parts of the ocean below 6,000 meters, in trenches where the pressure exceeds 1,000 atmospheres, scientists using deep-sea submersibles have discovered dense colonies of organisms living on the seafloor. Some of these communities exist at depths exceeding 9,500 meters, nearly 31,000 feet below the surface, in places where the pressure would instantly crush most equipment. These Hadal communities include tube worms, similar to Riftia pachyptila, but adapted to even more extreme pressures. There are fields of clams, some measuring 20 centimeters across, clustered in areas where methane and hydrogen sulfide seep from the seafloor. There are colonies of specialized bacteria forming thick mats on the sediment, and there are unusual crustaceans and worms that feed on the bacteria or on each other. The discovery of these deep-sea chemosynthetic ecosystems has profound implications. It demonstrates that life does not require sunlight, that entire complex communities can exist in complete darkness, powered entirely by chemical energy from the Earth's interior. It suggests that the range of habitable environments in the universe may be far greater than we once thought. If life can flourish at the bottom of Earth's oceans, in darkness, cold, and crushing pressure, then perhaps it could exist in the subsurface oceans of Europa, one of Jupiter's moons, or in the icy plumes of Enceladus, a moon of Saturn, both of which are believed to have liquid water beneath their frozen surfaces. The tube worms of the deep sea, with their symbiotic bacteria and their chemical-based existence, remind us that life is fundamentally about energy and chemistry, not about light and warmth. They show us that evolution can find solutions to survival in the most unlikely places, that the boundaries of habitability are not fixed but fluid, expanding wherever energy flows and chemistry allows. As you contemplate these creatures swaying in the darkness at the bottom of the ocean, sustained by volcanic gases and ancient bacterial partnerships, consider that they represent a form of life that might be more common in the universe than the sun-dependent ecosystems we are familiar with. Consider that they have been thriving in their alien environment for millions of years, following rhythms and strategies that we are only beginning to understand. Consider that they are proof that life, given energy and raw materials, can emerge in forms we would never have predicted. The living fossil of the twilight zone. There is a creature that inhabits the perpetual twilight of the mesopelagic zone, the layer of the ocean where the last faint traces of sunlight fade into absolute darkness. Its name is Vampiroteuthis infernalis, which translates literally to vampire squid from hell, a name that conjures images of predatory horror from the depths. But this name is misleading in almost every way. The vampire squid is not a vampire, it does not drink blood, and it is not even truly a squid. It is something far stranger and more fascinating, a living fossil that represents a unique branch of cephalopod evolution, and it has solved the problem of survival in one of the ocean's most hostile environments in a way unlike any other animal. The vampire squid lives in the oxygen minimum zone, a layer of the ocean where oxygen concentrations drop to levels that would suffocate most marine animals. This zone typically exists at depths between 600 and 1,200 meters, though its exact depth varies depending on location. In this region, oxygen saturation can drop to as low as 3% of surface levels. For comparison, most fish and active marine animals require oxygen levels of at least 20 to 30% to survive. The oxygen minimum zone is essentially a dead zone, a barrier that most oceanic life cannot cross. But Vampiroteuthis infernalis thrives there. It is the only cephalopod, the only member of the group that includes squid, octopuses, and cuttlefish, that can spend its entire life cycle in the oxygen minimum zone. And it does so through a suite of remarkable adaptations that make it one of the most specialized creatures in the deep sea. The vampire squid is small, typically reaching a maximum length of about 30 centimeters, with a gelatinous body that can appear velvety black or pale reddish depending on the lighting. Its most distinctive features are its eyes, which are proportionally the largest of any animal on Earth relative to body size. These enormous eyes, which can be blue or red depending on the light that strikes them, are adapted to detect the faintest bioluminescence in the darkness of the deep sea, allowing the vampire squid to spot potential prey, predators, or mates from a considerable distance. Between its 8 arms, which are connected by a webbing of dark skin, the vampire squid has something no other cephalopod possesses: 2 long retractile filaments. These filaments can extend to lengths much greater than the squid's body, and for years, scientists were puzzled about their function. The filaments are covered in tiny hair-like structures called cirri, and they are highly sensitive to touch and chemical signals in the water. Recent research has revealed that these filaments are feeding structures, and they are used in a manner completely unlike any other cephalopod. Because the vampire squid is not a predator, it does not hunt living prey. Instead, it is a detritivore, an eater of debris, and specifically it feeds on marine snow. Marine snow is the constant rain of organic particles that falls from the surface waters of the ocean toward the deep. It consists of dead plankton, fecal pellets from animals, discarded mucus structures from gelatinous creatures, molted exoskeletons, fragments of larger organisms, and bacterial aggregates. This material drifts downward through the water column, and in the oxygen minimum zone it becomes one of the primary food sources for animals that cannot actively hunt in the low oxygen conditions. The vampire squid extends one of its long filaments into the water and allows it to drift, collecting particles of marine snow that stick to the cirri along its length. After a period of collection which can last for minutes or hours, the squid retracts the filament and runs it through its arms, scraping off the accumulated debris and transferring it to its mouth. This feeding strategy is slow and methodical, perfectly suited to the vampire squid's low-energy lifestyle. Because energy conservation is crucial in the oxygen minimum zone, the vampire squid has evolved to have the lowest mass-specific metabolic rate of any cephalopod. It moves slowly, drifting more than swimming, using its fins to make gentle adjustments to its position. It does not engage in rapid bursts of speed or long-distance migrations. Everything about its physiology is optimized for minimal energy expenditure. Its blood contains a highly efficient form of hemocyanin, the copper-based molecule that cephalopods use to transport oxygen. This hemocyanin binds oxygen more effectively than that of other cephalopods, allowing the vampire squid to extract oxygen from water where other species would suffocate. Its gills have an unusually large surface area, maximizing the absorption of what little oxygen is available. The vampire squid's body is gelatinous and largely neutrally buoyant, meaning it requires very little effort to maintain its position in the water column. It has reduced musculature compared to more active cephalopods, trading strength and speed for efficiency. And it has sophisticated statocysts, balance organs similar to the inner ear in vertebrates, that allow it to orient itself in the darkness without expending energy on constant swimming. When threatened, the vampire squid has a unique defense strategy. It does not flee at high speed like other cephalopods. Instead, it inverts its arms and webbing, wrapping them around its body to form a protective cloak that exposes only the rough, spiny cirri on the outer surface of its arms. This posture, combined with its dark coloration, makes it difficult for predators to grasp. If further threatened, it can produce bioluminescent displays from light organs on the tips of its arms, creating flashing patterns that may confuse predators. And as a last resort, it can eject a cloud of bioluminescent mucus, a sticky, glowing substance that hangs in the water, potentially distracting a predator while the squid drifts away into the darkness. The evolutionary history of Vampiroteuthis infernalis is fascinating. It is the sole surviving member of the order Vampiromorpha, a group that dates back at least 120 million years and possibly much longer. Fossil evidence suggests that ancient vampiromorphs once lived in shallower waters, but at some point, likely in response to changing ocean conditions and the development of oxygen minimum zones during the Cretaceous and Cenozoic periods, they retreated to the deep sea. Modern vampire squids are adapted to these low-oxygen environments in ways their shallow-water ancestors were not, suggesting that the expansion of oxygen minimum zones created new ecological opportunities that the vampiromorphs were uniquely positioned to exploit. Today, vampire squids are found in temperate and tropical oceans around the world, always in the oxygen minimum zone. They occupy a niche that few other animals can access, feeding on a resource that is abundant but difficult to utilize, living in conditions that most predators cannot tolerate. This gives them a form of safety. In the oxygen minimum zone, there are few animals capable of hunting actively, and those that do venture into these depths typically cannot remain for long. But the vampire squid's reliance on the oxygen minimum zone also makes it potentially vulnerable to changes in ocean chemistry. As climate change affects ocean circulation and oxygen distribution, the depth and extent of oxygen minimum zones are shifting. In some regions, they are expanding as warming waters hold less dissolved oxygen. In other areas, changes in currents are altering their structure. How these changes will affect vampire squid populations is not yet clear, but it reminds us that even creatures as specialized and ancient as Vampiroteuthis infernalis are not immune to the rapid environmental changes occurring in our oceans. The vampire squid, despite its fearsome name, is a gentle creature, a scavenger that has found a way to survive in one of Earth's most challenging environments by being patient, efficient, and specialized. It represents a form of life shaped by constraints we can barely imagine, a testament to evolution's ability to find solutions even in the most oxygen-starved, lightless depths. And it reminds us that the ocean's twilight zone, the vast region between surface and abyss, is not empty but full of life forms. We are only beginning to understand creatures that have evolved strategies for survival that we would never have predicted. Giants in the abyss. In the deep ocean, there exists a phenomenon that seems to defy logic. In an environment where food is scarce, where energy must be conserved, where every calorie counts, some organisms have evolved to be enormous. This pattern is called abyssal gigantism or deep-sea gigantism, and it has produced creatures that dwarf their shallow water relatives, sometimes growing to sizes that seem impossible given the constraints of their environment. One of the most striking examples is the giant isopod, creatures in the genus Bathynomus that can reach lengths of up to 50 centimeters and weights exceeding 1.7 kilograms. To put this in perspective, the common pill bugs or roly-polies that you might find in your garden, creatures that are close relatives of giant isopods, are typically less than 2 centimeters long. The deep-sea versions are more than 20 times larger. Giant isopods inhabit the cold, dark regions of the ocean floor at depths ranging from 200 to 2,000 meters. They are scavengers crawling slowly across the sediment on their 14 legs, searching for food. Their diet consists primarily of dead and decaying organisms that sink from the waters above, including fish, squid, whales, and anything else that falls into their domain. They have powerful jaws that can tear through tough organic material, and they can consume large quantities of food when it is available. But food in the deep sea is unpredictable. A whale carcass might provide a feast that lasts for weeks, but months could pass between such windfalls. During these periods of scarcity, giant isopods can survive for extended periods without eating. Laboratory studies have documented individuals going more than 5 years without food, sustaining themselves on stored reserves. This ability to endure long fasts is crucial for survival in an environment where the next meal is never guaranteed. Why did these creatures evolve such large size? The answer is not entirely clear, but several theories have been proposed. One is Bergmann's rule, an ecological principle stating that within a species or a closely related group of species, individuals in colder climates tend to be larger than those in warmer climates. Larger body size offers several advantages in cold environments, including better heat retention and more efficient metabolism. The deep ocean is perpetually cold, typically between 2 and 4 degrees Celsius, and this constant cold may have favored the evolution of larger body sizes. Another factor may be the reduced pressure from predators in the deep sea. With fewer animals capable of hunting in the abyss, there may be less evolutionary pressure to remain small and inconspicuous. A larger body offers advantages in storing energy reserves, in covering more ground while searching for food, and in dominating resources when they are found. There is also the possibility that the high pressure of the deep sea itself plays a role. At depths of 2,000 meters, the pressure is approximately 200 times greater than at the surface. This pressure affects how gases dissolve in body fluids and how proteins fold and function. Some researchers have hypothesized that larger body size might be advantageous for managing the physiological challenges of extreme pressure, though this remains speculative. Whatever the causes, giant isopods are not alone in their gigantism. The deep sea is home to many other oversized species. There are giant squid that can reach lengths of 12 to 13 meters with eyes the size of dinner plates. There are colossal squid, even larger, with tentacles armed with rotating hooks. There are giant sea spiders with leg spans exceeding 70 centimeters. There are amphipods, shrimp-like crustaceans that grow to 30 centimeters in the deepest trenches. But gigantism is not the only unusual adaptation found in the deep sea. There are creatures with body plans that seem almost alien in their strangeness. Consider the barrel-eye fish, scientifically known as Macropinna microstoma. This fish, which lives at depths of 600 to 800 meters, has a transparent head. Its skull is a fluid-filled dome completely see-through, and through this transparent shield you can see the fish's barrel-shaped eyes pointing upward and even its brain. For years, specimens of barrel-eye fish were brought to the surface damaged, and scientists believed the eyes were fixed in the upward position. But in 2004, researchers from the Monterey Bay Aquarium Research Institute observed living barrel-eye fish in their natural habitat using remotely operated vehicles, and they discovered that the eyes can rotate. When the fish is searching for food drifting down from above, its eyes point upward. But when it spots prey and moves to intercept it, the eyes rotate forward, allowing the fish to see where it is going. The green coloration of the eyes may serve as a filter, helping the fish distinguish bioluminescent signals from the general downwelling light in the twilight zone. The transparent head protects the delicate eyes from the stinging tentacles of siphonophores, jellyfish-like creatures that the barrel-eye may steal food from. Then there are the anglerfish, perhaps the most iconic deep-sea creatures. Female anglerfish have a modified dorsal spine that extends forward over the mouth, tipped with a bioluminescent lure. This lure contains symbiotic bacteria that produce light, and the female anglerfish uses it to attract prey in the darkness. When a smaller fish or shrimp investigates the glowing lure, the anglerfish strikes with surprising speed, engulfing the prey in its enormous tooth-filled mouth. Male anglerfish are radically different from females. In many species, males are tiny, sometimes only a few centimeters long compared to females that can reach 30 centimeters or more. The male's sole purpose is reproduction, and in some species he accomplishes this by biting onto the female's body and fusing with her, becoming a permanent parasitic appendage that provides sperm when needed. Multiple males can attach to a single female, ensuring that when she is ready to spawn, fertilization can occur. This extreme sexual dimorphism and parasitic mating strategy may be an adaptation to the vastness and emptiness of the deep sea. Finding a mate in the darkness is difficult, and once found, ensuring that reproduction can occur is paramount. By physically fusing with the female, the male guarantees his genetic legacy and the female gains a permanent source of sperm without needing to search for mates. The frilled shark, Chlamydocylacus sanguineus, represents yet another form of deep-sea strangeness. This eel-like shark has changed little over millions of years, retaining primitive features that make it look like something from the age of dinosaurs. It has approximately 300 teeth arranged in 25 rows, each tooth having multiple needle-like cusps. These teeth are not for chewing, but for gripping slippery prey like squid and fish, which the frilled shark swallows whole. The frilled shark's gills are among its most distinctive features, with each of the 6 gill slits extending far down the body and edged with frilly-looking tissue, giving the shark its name. It lives at depths of 500 to 1,500 meters, though it can occasionally be found deeper or in shallower waters. Its body is flexible and serpentine, allowing it to navigate through the water with undulating movements, unlike the more rigid swimming of most sharks. These creatures, the giant isopods, barrel-eye fish, anglerfish, frilled sharks, and countless others, represent the ocean's experimentation with form and function under extreme conditions. They remind us that evolution is not a process that produces standardized results, but one that generates diversity. That the same challenges can be met with radically different solutions, and that the deep sea, far from being a barren wasteland, is a laboratory of biological innovation. The adaptations we see in these organisms are responses to specific environmental pressures: the scarcity of food, the absence of light, the crushing pressure, the cold, the need to find mates in a vast, dark space. Each species has solved these problems in its own way, and the result is a menagerie of creatures that seem fantastical, impossible, yet they are real, and they are thriving in the largest habitat on Earth, the deep ocean that covers more than 60% of our planet's surface and yet remains largely unexplored. Communities powered by Earth's heat. In the summer of 2025, an international team of scientists aboard the research vessel Falkor embarked on an expedition to explore seamounts and trenches in the Northwest Pacific Ocean. Using a remotely operated vehicle capable of descending more than 9,500 meters below the surface, they ventured into the Hadal Zone, the deepest layer of the ocean where pressures exceed 1,000 atmospheres and sunlight is a concept that has no meaning. What they found astonished even seasoned deep-sea researchers. At depths exceeding 31,000 feet, nearly 6 miles below the surface, in trenches where the seafloor creases and folds under the weight of colliding tectonic plates, they discovered thriving communities of life. There were dense colonies of tube worms, their white tubes clustered so tightly that they formed underwater meadows. There were fields of clams, bivalves with shells measuring 20 centimeters across, their tissues harboring symbiotic bacteria. There were mats of microorganisms coating the rocks in thick white layers. And moving through this landscape were unusual crustaceans, worms, and other creatures, many of which had never been seen before. These were chemosynthetic communities, ecosystems powered not by sunlight but by chemical energy seeping from the Earth's interior. And they represented a form of life that challenges our most basic assumptions about where organisms can exist and what resources they require to survive. Chemosynthetic communities were first discovered at hydrothermal vents in 1977, but since then they have been found in many other settings. In addition to hot vents where superheated water gushes from the seafloor, there are cold seeps where methane and hydrogen sulfide ooze from sediments. There are whale falls, the carcasses of dead whales that sink to the seafloor and create temporary oases of organic material and gases produced by decomposition. There are even subsurface communities living in the porous rock of the oceanic crust, far below the seafloor sediment. All of these environments have one thing in common. They provide chemical compounds that can be used as energy sources by specialized bacteria and archaea. These microorganisms perform chemosynthesis using the chemical energy released by reactions involving hydrogen sulfide, methane, hydrogen gas, ammonia, or other compounds to convert carbon dioxide into organic molecules. In this way, they fill the same ecological role that photosynthetic organisms like plants and algae fill in surface ecosystems. They are primary producers, the foundation of the food web, creating organic matter from inorganic precursors. But instead of using light energy, they use chemical energy. Instead of producing oxygen as a byproduct, they may produce sulfur, methane, or other compounds depending on the specific chemical reactions involved. And instead of being constrained to environments where sunlight penetrates, they can exist anywhere chemical energy is available. The diversity of chemosynthetic microorganisms is staggering. There are sulfur-oxidizing bacteria that thrive in the plumes of hydrothermal vents, harvesting energy from hydrogen sulfide and producing elemental sulfur as a waste product. There are methanogens, archaea, that produce methane by combining hydrogen gas with carbon dioxide. There are iron-oxidizing bacteria that extract energy from the transformation of ferrous iron to ferric iron. There are organisms that can use ammonia, nitrite, or even arsenate as energy sources. Many of these microorganisms live in symbiotic relationships with larger animals. The giant tube worms, Riftia pachyptila, harbor sulfur-oxidizing bacteria in their trophosome, as we discussed earlier. But they are not alone. There are clams, mussels, and snails that host chemosynthetic bacteria in specialized organs. There are shrimp that cultivate bacteria on their bodies. There are even fish that may harbor bacterial symbionts. These partnerships allow the animals to exploit chemical energy sources that they could not use on their own. The bacteria gain a stable environment and a constant supply of the chemicals they need. The host gains nutrition from the organic compounds the bacteria produce. It is a mutually beneficial relationship that has evolved independently in many different lineages, a testament to the power of chemosynthesis as a survival strategy. The recent discoveries in the Hadal trenches of the Pacific suggest that chemosynthetic communities may be more widespread than previously thought. The trenches are geologically active regions where tectonic plates converge, creating conditions conducive to the release of methane and hydrogen sulfide from the seafloor. The dense colonies of organisms found there indicate that these chemicals are abundant enough to support complex ecosystems even at the greatest depths of the ocean. Researchers have identified new species of tube worms, clams, and other organisms from these Hadal communities. Species that are adapted to pressures and conditions that exceed anything previously studied. Some of these organisms have enhanced pressure-resistant proteins, modified cellular membranes, and other adaptations that allow them to function under extreme compression. They represent the frontier of life's ability to colonize extreme environments. There is also increasing evidence that chemosynthetic communities may be widespread in the oceanic crust itself, far below the seafloor. Volcanic rock, particularly basalt, contains minerals that can react with seawater circulating through cracks and pores. These reactions produce hydrogen gas, which can be used by methanogens and other microorganisms. Some scientists estimate that the volume of the oceanic crust is so vast and the potential for these reactions so great that the subsurface biosphere may contain as much biomass as all the life in the ocean above. This subsurface realm is almost entirely unexplored. Drilling into the oceanic crust is technically challenging and expensive, and the microorganisms that live there are difficult to culture and study. But preliminary findings suggest that it is a thriving ecosystem, one that has existed for billions of years, powered by the slow, steady chemistry of rock and water. The existence of chemosynthetic ecosystems has profound implications for the search for life beyond Earth. If life can thrive in complete darkness in the absence of sunlight powered by chemical reactions involving simple compounds like hydrogen sulfide and methane, then the range of potentially habitable environments in the universe expands dramatically. Europa, one of Jupiter's largest moons, is covered in ice but is believed to have a liquid ocean beneath its frozen shell. This ocean is in contact with a rocky seafloor, and tidal forces from Jupiter's gravity may generate enough heat to drive volcanic activity and create conditions similar to Earth's hydrothermal vents. If that is the case, then Europa's ocean might host chemosynthetic life forms, analogous to those found at Earth's vents and seeps. Enceladus, a moon of Saturn, has geysers that shoot plumes of water vapor and organic compounds into space. These plumes appear to originate from a subsurface ocean, and the detection of hydrogen gas in the plumes suggests that there are chemical reactions occurring between the ocean and the rocky core of the moon. Reactions that could provide energy for chemosynthetic microorganisms. Even Mars, which is cold and dry today, may have subsurface environments where liquid water and chemical gradients exist, potentially supporting microbial life forms that use chemosynthesis to survive. The chemosynthetic communities of Earth's deep ocean are not just fascinating in their own right. They are windows into the possibilities of life in the universe, demonstrations that life does not require sunlight or temperate conditions or any of the things we once thought were essential. Life requires energy, raw materials, and liquid water. If those are present, evolution can find ways to make use of them, even in the harshest, darkest, most extreme environments imaginable. As you drift towards sleep, consider that beneath the ocean floor, in the cracks of volcanic rock, in the sediments of deep trenches, there are microorganisms quietly harvesting energy from the planet's interior, sustaining ecosystems that are utterly independent of the sun. Consider that life, given billions of years and the right chemistry, can colonize not just the surface of worlds, but their interiors, their deep places, their hidden realms. Consider that the universe may be full of such life, thriving in darkness, powered by the slow, patient chemistry of planets and moons we have not yet explored. The molecular secrets of survival. When scientists study organisms that live in extreme environments, whether in the crushing depths of the ocean, in the frozen wastes of Antarctica, or in the searing heat of volcanic vents, they are not just documenting unusual creatures. They are uncovering molecular strategies, biochemical innovations that allow life to persist where it should be impossible. And these discoveries are revealing fundamental principles about the nature of life itself. One of the most important insights has come from studying DNA repair mechanisms. All organisms face the problem of DNA damage. Cosmic radiation, ultraviolet light, reactive oxygen species, and simple errors during DNA replication can cause breaks in the DNA strands, mutations in the genetic code, and other forms of damage. If left unrepaired, this damage accumulates over time, leading to cancer, aging, and eventually death. Most organisms have DNA repair systems, molecular machinery that can detect and fix damaged DNA. But organisms in extreme environments have evolved particularly sophisticated repair mechanisms. Tardigrades, as we discussed earlier, produce the DSUP protein that shields DNA from damage in the first place. But they also have highly efficient repair enzymes that can quickly fix any damage that does occur. Some bacteria that live in highly radioactive environments, such as Deinococcus radiodurans, can survive radiation doses thousands of times greater than what would kill a human. These bacteria have multiple copies of their genome, So if radiation shatters the DNA in one copy, they can use the intact copies as templates to repair the damaged one. They also have exceptionally efficient DNA repair enzymes and protective compounds that neutralize reactive oxygen species before they can cause damage. The study of these repair mechanisms has practical applications. Cancer treatment often involves radiation or chemotherapy drugs that work by damaging the DNA of cancer cells. But these treatments also damage healthy cells, causing side effects. Understanding how organisms like tardigrades and radiation-resistant bacteria protect and repair their DNA could lead to therapies that protect healthy tissues during cancer treatment, or drugs that enhance the body's own repair mechanisms to treat genetic diseases. Another fascinating area of research involves antifreeze proteins. Many organisms that live in cold environments, whether in polar seas or at high altitudes, face the problem of ice formation in their tissues. When water freezes, it forms crystals that can puncture cell membranes and disrupt cellular structures. Most organisms cannot survive this. But some fish, insects, and other organisms have evolved antifreeze proteins, molecules that interfere with the formation of ice crystals. These proteins bind to tiny ice crystals as they begin to form, preventing them from growing larger. They can lower the freezing point of body fluids by several degrees, or in some cases allow the organism to tolerate ice formation in certain tissues while keeping critical organs ice-free. The Antarctic ice fish, for example, produces multiple types of antifreeze glycoproteins that allow it to swim in water that is below the normal freezing point of fish blood. Some Arctic insects can survive being frozen solid with antifreeze proteins protecting their cells from damage. The mechanisms by which these proteins work are still being studied, but they have potential applications in cryopreservation, organ transplantation, and even in developing crops that can withstand frost. Bioluminescence, the ability to produce light through chemical reactions, is another molecular innovation that is widespread in the deep sea. More than 90% of deep-sea organisms are estimated to have some form of bioluminescence. They use it for a variety of purposes: to attract prey, to communicate with potential mates, to startle or confuse predators, or to camouflage themselves by matching the faint downwelling light from above. The chemistry of bioluminescence involves a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferase catalyzes the oxidation of luciferin, light is produced as a byproduct. Different organisms use different types of luciferin and luciferase, and they have evolved ways to control when and where light is produced, often by regulating the availability of oxygen or other cofactors. Understanding bioluminescence has led to important biotechnological applications. Luciferase from fireflies is now used as a reporter gene in molecular biology, allowing researchers to track gene expression in cellular processes by measuring light production. Bioluminescent proteins from jellyfish, particularly green fluorescent protein, have revolutionized cell biology by allowing scientists to visualize proteins and cellular structures in living cells. There is also the phenomenon of extremozymes, enzymes that function in extreme conditions. Most enzymes are proteins that catalyze chemical reactions in cells, and they typically work best at moderate temperatures near neutral pH and normal pressures. But enzymes from extremophiles can function at boiling temperatures, in highly acidic or alkaline conditions, or under extreme pressures. Taq polymerase, an enzyme isolated from the bacterium Thermus aquaticus, which lives in hot springs, is stable at high temperatures and is used in the polymerase chain reaction. a technique that revolutionized molecular biology by allowing scientists to amplify small amounts of DNA. Other extremozymes are used in industrial processes, in detergents that work at high temperatures, in biofuels production, and in various other applications. The study of extreme organisms has also revealed unexpected examples of convergent evolution, where unrelated organisms evolve similar solutions to similar problems. The symbiosis between animals and chemosynthetic bacteria, for example, has evolved independently in tube worms, clams, mussels, snails, and other groups. The use of antifreeze proteins has evolved separately in fish, insects, plants, and bacteria. Bioluminescence has arisen independently in many different lineages, from bacteria to fungi to insects to fish. This convergence suggests that there are only so many ways to solve certain biological problems, and that evolution, given similar challenges, tends to arrive at similar solutions. It also suggests that if we were to find life on other planets, it might have features recognizable to us, not because of common ancestry, but because of common physics and chemistry. At the molecular level, life in extreme environments teaches us that the machinery of biology is more flexible, more adaptable than we often assume. Proteins can be re-engineered by evolution to function at temperatures that would normally denature them. Membranes can be modified to remain fluid in freezing conditions or stable under extreme pressure. Metabolic pathways can be rerouted to use different energy sources or to produce different end products. This adaptability is a testament to the power of natural selection acting over long periods of time, testing countless variations and retaining those that work. It is also a reminder that the conditions we consider normal, the temperature range we find comfortable, the oxygen concentration we require, are not universal requirements for life. They are simply the conditions that our particular branch of the tree of life happens to be adapted to. There are other branches of that tree, branches that have adapted to conditions we find extreme, and in their molecular machinery, in the proteins and pathways they use to survive, there are lessons about the fundamental nature of life, about its limits and its possibilities, about what is essential and what is merely contingent. Implications for life beyond Earth. The discovery of life in Earth's most extreme environments has fundamentally altered how we think about the search for life elsewhere in the universe. For much of human history, we assumed that life required conditions similar to those on Earth's surface: moderate temperatures, liquid water, sunlight, and an oxygen-rich atmosphere. The discovery of organisms thriving in deep-sea vents, in acidic hot springs, in the frozen wastes of Antarctica, and in other extreme environments has shown that these assumptions were too narrow. Life, it turns out, is far more adaptable than we imagined. It can survive and even thrive in conditions that we once thought were incompatible with any form of biological organization. And this realization has expanded the list of places in our solar system and beyond where we might find living organisms. Europa, one of Jupiter's moons, is now considered one of the most promising locations to search for extraterrestrial life. Beneath its ice-covered surface, Europa is believed to have a global ocean of liquid water, perhaps 100 kilometers deep. This ocean is in contact with a rocky seafloor, and the tidal forces exerted by Jupiter's gravity likely generate enough heat to drive geological activity. If there are hydrothermal vents on Europa's seafloor similar to those found on Earth, then the conditions for chemosynthetic life may exist. The chemistry would be familiar: water, rock, heat, and the potential for chemical gradients. The organisms, if they exist, might be microbial, similar to the bacteria and archaea that live at Earth's vents. Or they might be more complex, analogous to the tube worms, clams, and other animals that form chemosynthetic communities. We simply do not know. But the existence of such organisms is not a wild speculation. It is a reasonable hypothesis based on what we have learned from Earth's deep-sea ecosystems. Enceladus, a small moon of Saturn, has become another focus of astrobiological interest. In 2005, the Cassini spacecraft discovered that Enceladus has geysers erupting from its south polar region, shooting plumes of water vapor and ice particles into space. Analysis of these plumes revealed the presence of organic compounds, salt, and hydrogen gas. The hydrogen is particularly significant because it suggests that there are chemical reactions occurring between water and rock. Reactions that produce energy that could support chemosynthetic microorganisms. The fact that we can sample the contents of Enceladus's subsurface ocean by flying through its plumes makes it an especially attractive target for the search for life. We do not need to land on the surface or drill through kilometers of ice. We can collect material directly from the ocean and analyze it for signs of biological activity. Mars, our planetary neighbor, was once much wetter and warmer than it is today. Billions of years ago, it had rivers, lakes, and possibly even an ocean. But its atmosphere thinned, its surface water evaporated or froze, and it became the cold, dry desert we see today. However, there is evidence that liquid water may still exist in the Martian subsurface, in aquifers or pockets of brine kept liquid by dissolved salts and geothermal heat. If such environments exist and if they are in contact with minerals that can provide chemical energy, then microbial life might persist there deep below the surface, surviving in a refuge similar to the subsurface biosphere in Earth's oceanic crust. Beyond our solar system, the search for habitable exoplanets has focused primarily on worlds in the habitable zone of their stars, the region where temperatures are right for liquid water to exist on the surface. But the discovery of chemosynthetic ecosystems on Earth suggests that subsurface habitability may be more important than surface conditions. A planet or moon does not need to be in the habitable zone to have liquid water and chemical energy gradients in its interior. It just needs to have enough geological activity to drive those processes. This vastly expands the number of potentially habitable worlds. Moons orbiting gas giants even far from their stars could have subsurface oceans heated by tidal forces. Small, rocky planets with thick ice shells could harbor liquid water beneath. Rogue planets, drifting through interstellar space with no star at all, could potentially have subsurface biospheres powered by radioactive decay and residual heat from their formation. The study of extreme life on Earth has also informed the development of techniques to detect life elsewhere. Biosignatures, the chemical or physical signs that life is present, can take many forms. On Earth's surface, the presence of oxygen in the atmosphere is a biosignature, a sign that photosynthetic organisms are producing it. But in subsurface or deep-sea environments, other biosignatures are more relevant. The presence of specific organic molecules, unusual isotopic ratios in carbon or sulfur that suggest biological processing, or disequilibrium in chemical concentrations that could indicate metabolic activity. Instruments are being developed to detect these biosignatures in samples from other worlds. Mass spectrometers can identify organic molecules. Isotope ratio analyzers can look for the subtle chemical signatures of biological processes. Microscopes can search for cell-like structures or other morphological signs of life. But perhaps the most important lesson from studying extreme life on Earth is that we need to be open-minded about what life might look like elsewhere. The organisms we have found in extreme environments are often radically different from those in more familiar habitats. They use different energy sources, different metabolic pathways, different survival strategies. Some of them challenge our definitions of what it means to be alive. If we find life on Europa or Enceladus or Mars, it will almost certainly be different from life on Earth. It may use different biochemistry, different types of molecules to store information or catalyze reactions. It may have a different genetic code or no genetic code as we understand it. It may exist in forms we do not immediately recognize as living. This is why the study of extremophiles is so important. They expand our imagination. They show us the breadth of possibilities that life can explore. And they prepare us for the day when we might encounter life that evolved independently, under different conditions, following different rules. The implications extend beyond the search for extraterrestrial life. Understanding how organisms survive in extreme environments has practical applications here on Earth. The proteins and mechanisms that allow tardigrades to survive desiccation could improve methods for preserving vaccines, blood and organs for transplantation. The DNA repair systems of radiation-resistant bacteria could lead to new treatments for cancer and genetic diseases. The enzymes from heat-loving microorganisms are already used in industrial processes and biotechnology. As we face environmental challenges on Earth, from climate change to pollution to habitat destruction, the resilience of extremophiles offers both hope and perspective. These organisms have survived mass extinctions, dramatic climate shifts, and geological upheavals over billions of years. They demonstrate that life, at least microbial life, is remarkably robust. But they also remind us that the familiar ecosystems we depend on, with their complex food webs and delicate balances, are more fragile, and that the loss of biodiversity we are causing is a loss not just of species but of evolutionary possibilities, of solutions to problems we have not yet encountered. The enduring mystery of the deep. As we reach the end of our journey through the impossible organisms of the ocean, it is worth reflecting on how much remains unknown. The deep sea, despite being the largest habitat on Earth, is less explored than the surface of the Moon or Mars. We have mapped only a small fraction of the seafloor in detail. We have visited an even smaller fraction with submersibles or remotely operated vehicles. And the organisms we have cataloged represent, by most estimates, only a tiny percentage of the species that actually exist in the deep. Every expedition to the deep ocean discovers new species, sometimes dozens of new species, sometimes hundreds. The recent expedition to the Pacific trenches found more than 100 potentially new species in just a few weeks of exploration. And these were just the animals and larger organisms that could be seen and collected. The microbial diversity, the viruses and bacteria and archaea that inhabit the deep sea and the subsurface biosphere is largely unmapped and unstudied. We are still discovering fundamentally new types of ecosystems. The chemosynthetic communities at hydrothermal vents were unknown until 1977. Cold seeps were discovered later. Whale falls as a distinct ecosystem were recognized in the 1980s. Subsurface communities in the oceanic crust are only beginning to be explored now. How many other ecosystems are waiting to be found? How many other ways has life found to survive in the ocean's depths? There are vast regions of the ocean that have never been explored. The Hadal Zone, the deepest parts of the ocean trenches, has been visited only a handful of times. The abyssal plains, the flat stretches of seafloor that cover much of the deep ocean, are poorly studied. The midwater, the vast three-dimensional space between the surface and the bottom, is almost entirely unexplored despite being the largest living space on the planet. New technologies are beginning to change this. Autonomous underwater vehicles can explore the deep without human pilots, covering larger areas and staying down longer. Sensors are becoming more sophisticated, able to detect chemical signatures, measure metabolic activity, and even sequence DNA from environmental samples. Satellite technology is improving our ability to map the seafloor from above. But even with these advances, we are still just scratching the surface of understanding the deep ocean's biodiversity and its role in the planet's systems. The organisms we have encountered in this journey, the immortal jellyfish, the tardigrades, the tube worms, the vampire squid, the giants of the abyss, the chemosynthetic communities, all remind us of the incredible creativity of evolution. Given time and challenge, natural selection can produce solutions that seem impossible. Organisms that thrive in conditions that would kill most life. Strategies for survival that defy our expectations. These creatures are not anomalies or curiosities. They are representatives of successful lineages that have been refining their adaptations for millions of years. They are proof that the conditions we consider normal are just one narrow band in a much broader spectrum of possibilities. They are evidence that life is not fragile and delicate, but robust and persistent, able to colonize every niche where energy and raw materials are available. As you settle into sleep, let your mind drift through the dark waters of the deep ocean. Picture the tube worms swaying in the currents above a hydrothermal vent, sustained by bacteria that harvest energy from the Earth's interior. Picture the vampire squid drifting in the oxygen-starved twilight, patiently collecting the debris that falls from above. Picture the tardigrades in the moss outside your window, carrying within their microscopic bodies the ability to survive the vacuum of space and the fires of stellar radiation. Picture the immortal jellyfish pulsing through the water, holding the secret to reversing time itself. These organisms exist. They are real. They are part of the same tree of life that includes us, shaped by the same processes of variation and selection, constructed from the same basic chemistry. But they have taken that chemistry to extremes we are only beginning to understand, and they have solved problems that our technology cannot yet match. The ocean is vast and deep and dark, and it holds mysteries we have not yet imagined. But what we have learned so far from the creatures that shouldn't exist but do is that life is more adaptable, more resilient, more creative than we ever thought possible. And that wherever in the universe there is energy and chemistry and liquid water, there might be life, finding ways to survive and thrive in forms we cannot yet predict. Thank you for joining this exploration of the impossible. If these quiet journeys through the mysteries of science bring you peace, consider subscribing to the channel or leaving a like. It helps others find these gentle paths through knowledge and wonder. Rest well, knowing that the ocean continues its slow, patient work, harboring life in the darkness, keeping secrets in the cold, proving with every organism that exists in impossible places that the boundaries of possibility are always farther than we imagine. Sleep well.